Sample preparation is one of the most variable and time-consuming steps in the analysis of proteins by mass spectrometry, and the quality and reproducibility of protein extraction, digestion, and preparation significantly impact the results. Typical protein sample preparation for mass spec analysis is difficult, time-consuming, and tedious, and can take up to 2 days to complete. The Thermo EasyPep Mini MS Sample Prep Kit enables efficient and reproducible processing of cultured mammalian cells and tissues for proteomic mass spec analysis in 2 to 4 hours. The kit contains pre-formulated buffers, a mass spec grade trypsin lyse-C enzyme mix, peptide cleanup columns, and an optimized protocol to generate 10 to 100 micrograms of mass spec compatible peptide samples. Here we describe the protocol for the EasyPep Mini MS Sample Prep Kit. The first stage of the sample preparation is the chemical and enzymatic processing of the sample. For cultured cells, add 100 microliters of lysis buffer and 1 microliter of universal nuclease to a minimum of 1 million cells. Pipette up and down for 10 to 15 cycles until sample viscosity is reduced. For tissue samples, add 100 microliters of lysis solution and 1 microliter of universal nuclease per 5 milligrams of tissue. Disrupt the sample with a tissue homogenizer until it is homogenized. Centrifuge tissue lysates at 16,000 G for 10 minutes and collect supernatant. Determine the protein concentration of the cell or tissue supernatant using established methods, such as the Thermo Pierce Rapid Gold BCA Protein Assay Kit. Transfer 10 to 100 micrograms of protein sample into a new microcentrifuge tube. Adjust the final volume to 100 microliters with lysis solution. Add 50 microliters of reduction solution to the sample. Gently mix by tapping the side of the tube. Add 50 microliters of alkylation solution to the sample and repeat the gentle mixing. Incubate the sample at 95 degrees Celsius using a heat block for 10 minutes to reduce and alkylate the protein sample. Remove the reduced and alkylated protein sample solution from the heat block and allow to cool to room temperature. Add 500 microliters of enzyme reconstitution solution to 1 vial of trypsin lyse-C protease mix. Add 50 microliters of this enzyme solution to the reduced and alkylated protein sample. Incubate at 37 degrees Celsius for 1 to 3 hours, with shaking to digest the protein sample. After incubation is complete, add 50 microliters of digestion stop solution to the sample. The second stage of sample preparation is the peptide cleanup. Remove the white plug at the bottom of the peptide cleanup column, loosen the top cap, and place the column into a 2 ml microcentrifuge tube. Centrifuge at 3000 G for 2 minutes to remove all liquid from the column. Discard the flow-through. Transfer the protein digest sample containing about 300 microliters total volume into the dry peptide cleanup column. Centrifuge at 1500 G for 2 minutes. Discard the flow-through. Add 300 microliters of the wash solution A into the column. Centrifuge at 1500 G for 2 minutes. Discard the flow-through. Add 300 microliters of the wash solution B into the column. Centrifuge at 1500 G for 2 minutes. Discard the flow-through. Transfer the peptide cleanup column into a new 2 ml microcentrifuge tube. Add 300 microliters of the elution solution into the column. Centrifuge at 1500 G for 2 minutes to collect the clean peptide sample. Dry the peptide sample using a vacuum centrifuge. Resuspend the sample in 100 microliters of 0.1% formic acid in water for LC-MS analysis. Determine peptide yield and concentration using a quantitative peptide assay. Adjust the peptide concentration with 0.1% formic acid in water for optimal LC-MS column loading. EasyPep Mini MS Sample Prep Kit reduces the standard protocol from 2 days to 2 to 4 hours. This kit includes a universal nuclease to reduce the viscosity caused by DNA liberated during lysis, eliminating the need for sonication. The protocol has a rapid combined reduction alkylation step for cysteine modification. The trypsin lyse-C protease mix enables faster, more complete digestion. The peptide cleanup columns and buffers are included in the kit to prepare detergent-free peptide samples for mass spec analysis. With the EasyPep Mini MS Sample Prep Kit, you can obtain comparable protein identifications and higher peptide yields in less time and with fewer steps than lab-made reagents or kits from other suppliers. Find out more about the fast and reproducible EasyPep Mini MS Sample Prep Kit and other related products at thermofisher.com/EasyPep.